Ruairi Robertson is a postdoctoral research fellow in the Blizard Institute, Queen Mary University of London. His research focuses on the influence of the gut microbiome in early life growth in children suffering from chronic and severe acute malnutrition in Zimbabwe and Zambia. Today, he'll be presenting the lecture, Malnourished Microbes, the Influence of the Gut Microbiome on Child Undernutrition. Ladies and gentlemen, please welcome Dr. Ruairi Robertson.
Hi everyone, thanks very much for having me. As discussed, I want to give a little kind of story to give a little background to my research, which gives a little introduction to the microbiome. In 1892, there was a big cholera epidemic which was spreading throughout the world, very much like COVID today. It started off in Asia and moved around to North Africa and made its way to Europe, where there was a guy in Paris, Elie Menschnikov, a very smart Russian scientist who would later win the Nobel Prize for discovering the role of phagocytes in the body. But he noticed that cholera was affecting some people, making them sick, and others seemed to get infected but not get really ill. So he conducted an interesting experiment where he decided to drink a broth of cholera himself to see what happens. And surprisingly, he didn't get sick. So he, like any kind of good scientist, wanted to increase the sample size and recruited a colleague to do the same thing. And this guy didn't get sick either, but he recruited a second colleague to drink this broth of cholera. And this guy got very sick and very nearly died. So Menschnikov decided to study cholera under the microscope and see how it interacted with other microbes from the human intestine. And what he found was that certain microorganisms appeared to stimulate the growth of cholera, whereas other microorganisms seemed to inhibit its growth. So he subsequently concluded that having the right balance of good to bad microorganisms in your intestine is necessary to help stave off disease. And he suggested and proposed that this mightn't only be the case for infectious diseases such as cholera, but could also play a role in longevity and staving off other forms of chronic disease as well. And he was ridiculed for this later in life. He identified this group of people in Eastern Europe who were living very long lives for that time. And he noted that they all drank this fermented milk every day. And this fermented milk has since been studied a bit further, and it's been isolated, or the bugs inside of it have been isolated. One of them was Lactobacillus bulgaricus, which was named after where it was discovered in Bulgaria. Now, this Lactobacillus bulgaricus is a common strain of lactobacilli, which is found in nearly every yogurt that you buy on the shelf these days. So Menschnikov's ideas were that by fostering the growth of healthy microorganisms in the intestines would help to live longer and stave off disease. But his ideas seem to be kind of lost in time because he died just before antibiotics were discovered. And since then, the kind of theory of medical practice was to fight off all forms of bacteria and microorganisms because we know that infectious diseases are potentially deadly. But since that time, in recent years, there's been kind of a re-emergence of this idea of Menschnikov that the right balance of commensal microorganisms in the gut is actually important, and we can't just have this model of every microorganisms being pathogenic or infectious. So that's what leads me to the human microbiome, which is what I study. And we are essentially ecosystems, our bodies. We contain trillions and trillions of microorganisms, from bacteria to fungi and to archaea and everything in between. And it's only in recent years, through advances in DNA sequencing technologies, that we've managed to appreciate the extent and the diversity of the human microbiome. So in the human body, we have very roughly 30 trillion mammalian cells. But if we count up just the bacterial cells that we are home to, we are outnumbered. It's roughly 40 trillion. And that doesn't include viruses or fungi or other forms of microorganisms. If we look at the kind of genetic potential of the human microbiome, we're actually outweighed even more. We're only about 1% human. There are 23,000 genes in the human genome, but there are a couple of million microbial genes in our body. So there's huge potential for this functional interaction between microorganisms and the human body, which could have a hugely positive or a negative effect on human health in so many ways. So the gut is the kind of most dense population of the microbiome in the human body, and that's because of the natural kind of warm environment, nutrient-rich, lacking oxygen, and so this creates an environment for a lot of these anaerobic and species to grow. And so this is kind of represented here in a scanning electron microscopy image of representing the diversity of the microbes from a human stool sample. And we can see here there's a huge number of different species here, which in this case are degrading a piece of dietary fiber. And these interact with the different parts of the intestine in many different ways, and with the underlying immune system to affect the body in a huge number of ways. So if we see that from an imaging of mouse colon, we can see here that a majority of the human microbiome resides in the lumen, where there's a big diversity of microorganisms. But if you see, there is certain ecological niches even within the gut itself where different microorganisms can grow and colonize. So we can see within the crypts of the colon here, there are certain species which lie within the mucosal layer and lie in really close interaction with the colonic epithelium. And these then interact with the underlying immune cells in the lamina propria, which can interact to recognize if these are commensal, recognize if these are pathogenic. And so there really is this huge two-way interaction between the microbiome and between the host at this mucosal barrier site in the gut, which can affect the gut in a huge number of ways. So at this kind of interface of the gut microbiome and the human immune system, this can lead to a number of downstream pathways affecting human health in so many ways that can't be summarized in one slide. But if we think that the human microbiome in the gut interacts with everything that we consume, be it medicines or food, they are essentially the gatekeepers to digest nutrients or to allow them into the body. Or there's even huge interesting studies showing how the makeup of the gut microbiome can affect the breakdown of drugs as well, and the efficacy of certain oral drugs. And of course, then there's even fascinating insights into how the gut microbiome interacts with the peripheral nervous system to affect functioning of the central nervous system and brain health. So there really is this huge potential of this microbial organ in the gut to interact with the host in so many ways that are only being discovered right now in recent years. And just to go full circle on that, this recent paper just a couple of months ago in Cell kind of really drove home what Menschnikov had proposed all those years ago, about 100 years ago. And this paper showed that the variability in an individual's gut microbiome really strongly affects the susceptibility or resistance to cholera infection. So it's nice to see that this old theory from 100 years ago is really coming through into modern day science, and that our gut microbiome can really influence our susceptibility not only to infectious diseases, but also to kind of modern chronic diseases as well. Now, my research is interested in the early phases of life and our initial interaction with the microbial world. Humans are essentially sterile in the womb, unless the mother has some sort of urogenital infection. Sometimes there can be some sort of interaction there, but in general, humans are sterile in the womb. And so then at birth, we're coated for the first time with this huge diversity of microbial life. And so this can have huge implications for the growth and the development of a child in early life, because we know that the human immune system is untrained in early life, that there are a lot of interacting pathways that happen in those first months of life, which can determine the trajectories of growth and development and of health into later life. So, I lost the slide. So in early life, the gut microbiome is quite kind of flexible, and it changes quite rapidly in this period known as the first 1,000 days. So the first 1,000 days are determined by the point of conception to about 2 years of life. And if you add up those days, it's about 1,000 days. So during that period, the gut microbiome changes quite rapidly. So we can see that in the first 6 months of life, the gut microbiome is made up of species which break down the sugars in breast milk. So we can see that here, these bifidobacteria or bacteroides are quite abundant in the first 6 months of life. But then after that, from about 6 months onwards, we can see that the gut microbiome changes to that of species which are more interested in degrading the sugars, or sorry, the polysaccharides from plants. So, can I get the next slide if that's possible? Great. We can see that here, that there's this kind of interaction in the first 2 years of life where the microbiome is quite flexible and changes. But then if we move to the next slide, we can see that from 2 or 3 years onwards, we can see, the next one, that the microbiome remains quite stable after that. So from 2 or 3 years onwards, the gut microbiome of a 2 or 3-year-old looks very much like that of an adult. And so what we know from this early life period is that this kind of flexibility of the microbiome in the first 2 years of life can determine maybe the kind of growth and developmental trajectories of a child later on. And so because our immune systems are kind of developing in that kind of flexible sense as well in early life, the gut microbiome may interact with some of these pathways. Next, please. Can I get the next slide, please? Ooh. Great. So if we flick onto the next slide again, my interest is in child malnutrition, and this is the most common form of malnutrition is child stunting. So a quarter of children around the world roughly are stunted, and that means when you have a length for age Z-score less than 2 standard deviations from the global norm. And this can have a huge impact on the health of the child into later life, because we know that in the first 1,000 days, the trajectories of growth can define your nutritional status in later life. So a child that is stunted up to 2 years, 3 years, tends to remain stunted throughout childhood and later on. And this can have huge implications for cognitive development, for susceptibility to infections, and so forth. So if we go on to the next slide, we can also see about severe acute malnutrition, which is the more classical form of malnutrition. And it comes in 2 forms, edematous and non-edematous. So if we see from the next slide, the form of that's characterized by a weight per height Z-score. And on the left, we can see is the non-edematous form, which is the more classical form of child malnutrition, where there's a lot of muscle and fat wasting. On the right is this edematous form of severe acute malnutrition, which is still quite a mysterious disorder, where you get this edema in the limbs of children, you get these kind of dermatosis phenomena, as you can see here, and lots of other phenomena that kind of distinguish it quite a lot from classical SAM, and yet these 2 conditions are treated exactly the same. So if we move on to the next slide, and again as well, we can see that malnutrition is a really still a complex problem throughout the world. It's still a very common problem. As I said, 1/5 to 1/4 of children around the world are still stunted, and there's still 47 million people around the world who are severely acute malnourished. So if we move to the next slide, and again, the kind of dogma of malnutrition is that it is completely defined by nutritional status or by diet, where, however, it's estimated that even if the most effective nutritional treatments were scaled up to a 90% coverage around the world, this would only reduce about 20% of stunting and 60% of wasting. So this really kind of drives home the fact that malnutrition ironically isn't just about nutrition and diet, and there might be some sort of intestinal or microbial effect. And what is evident in a lot of children, most children who are malnourished from these low resource settings, is this phenomenon of environmental enteric dysfunction or environmental enteropathy. We can see that on the left here of a healthy small intestine. You have the normal villi, projections, and a healthy looking ileum. Where on the right here, we can see this is environmental enteropathy. There's accumulation of immune cells in the lamina propria, there's blunted villi, a lot of inflammation. And so this can impair nutrient absorption, it can lead to inflammation, it can lead to intestinal infection. And this is what we presume kind of impairs the processes of growth. So next slide, please. And yes, so we have some evidence that there is some sort of microbial influence on child growth, even in the absence of infection. This was a meta-analysis, which was conducted by a colleague of mine a few years ago, which showed that even in the absence of a confirmed infection in low resource settings, antibiotics can actually help to increase growth in children in early life. So this would suggest that there might be some sort of dysbiotic gut microbiome or some sort of maybe slightly pro-inflammatory gut microbiome in the intestine, in kind of not including infection or pathogens, which might be impairing growth, and the antibiotics can somehow recover the microbiome in some sort of state to improve the pathways that lead to growth. Next slide, please. So we can see that from animal models as well. If you take this combination of commensal microorganisms, which are on the right, not overt pathogens, and combine them with a malnourished diet, on the fourth kind of picture on the right here, we can see this phenomenon of environmental enteropathy can be replicated in animals. You have shedding of the epithelial cells, inflammation, blunted villi. And so this isn't like an acute infection that these animals have. This is just a combination of kind of dysbiotic gut microbiome in a combination with an undernourished diet that can lead to this an enteropathy, which is associated with poor growth. Next slide, please. And we can see that from animal models, other animal models as well. So on the left is a mouse that's germ-free, so it's been reared with no exposure ever to microorganisms, reared in a bubble, and on the right is a regular wild-type animal. So if we move to the next slide, we can see that the growth is recovered in these animals when you colonize them with a single Lactobacillus plantarum species. Now, this is just a regular commensal organism, potentially probiotic, if you want to use that word, which can induce some of these pathways associated with IGF-1, insulin-like growth factor 1, or growth hormone, to stimulate growth in these animals. And that's just one microorganism on its own, which is having that growth recuperating effect in experimental animals. Next slide, please. So in humans, the best evidence from this is a study from Bangladesh from a few years ago, where the gut microbiome was sampled in children every month for the first 2 years of life. And this was in healthy growing children, but from a low resource setting. If we move to the next slide, we can see that the authors found 24 microbial species in the guts of these kids, which were most predictive of healthy growth. And they used this in an AI model to create a score of microbiota maturity. So if we move to the next slide, you can see that this score they call the Microbiota for Age Z-score. And so this is to be used in conjunction with a weight for height Z-score or a length for age Z-score as a predictor, as a gut microbiome predictor of growth and development in children. So if we move to the next slide, we can see how that works in a clinical setting. On the next slide, please. Great. So at enrollment, kids who have severe acute malnutrition, the red bar here, you can see that their microbiota for age Z-score is less than -5. So healthy would be around the 0 mark. So it's around -5, -6. And as they're treated in the orange bars there and the gray bars, their microbiota for age Z-score tends to recover. And this happens in conjunction with the recovery of their growth as well. However, at around 3 to 4 months, the microbiota for age Z-score tends to relapse again and goes back down again. And what we predict is that this might be a kind of predictive marker of relapse of severe acute malnutrition as well, or poor growth, which happens quite often in these children. Next slide, please. So we wanted to look at this in an observational study in Zimbabwe and Zambia over the last couple of years. So we move to the next slide. We recruited over 750 cases of severe acute malnutrition in 3 hospital sites in Zimbabwe and Zambia, and collected data from these children every few weeks after discharge in those that survived. And in a smaller number of these, we collected samples, stool samples, gastric fluid samples, plasma, and urine. And over the last couple of years, I've been kind of sequencing the microbiome from these children and conducting metabolomics on these kids to see if the gut microbiome is associated with their growth, is associated with mortality, etc. So we can see some results from that. If we move to the next slide, if we just plot these, each individual here is a dot on this PCA. And on the left, we have the SAM cases, and on the right, we have the controls. So just from the outlook, we can see that their gut microbiome is very different between these two, which we would expect. If we move to the next slide, we can see how this changes over time. On the left is the cases at baseline, and on the right in the purple is the healthy controls. You can see as these kids recover over time, up to about a year past recovery, their microbiome starts to look much more like a healthy child. So we're getting closer to maybe using this as a marker of predictive recovery. And we're working on this right now to see if this gut microbiome throughout recovery can be used to predict growth and predict maybe mortality as well. So we can move that to the next slide. And we can see how that works with the diversity of the microbiome as well. And this takes into account the number of species that are there and the number of kind of the distribution of microbes. We can see that on the right are the healthy controls and the left is baseline. And over time, the gut microbiome and the diversity tends to get back to a healthier state. Next slide, please. So finally, just looking what's there, if we move to the next slide again, we can see that at baseline, there's a huge number of E. coli and Klebsiella in these kids during hospitalization. And the relative proportion of these potentially pathogenic species tends to reduce over time to get back to that which looks more like a healthy control. So that's interesting to see that these severely acute malnourished kids have a kind of microbiome which is potentially pathogenic. Some of them might have infections ongoing, but over time this kind of tends to decline as these other kind of more commensal microorganisms take over. Next slide, please. So finally, if we look at kind of a clinical perspective of this, what is happening in the kind of clinical world? Well, there's been a few trials of probiotics in these cases of severe acute malnutrition. And the biggest trial of those, which was conducted a few years ago, failed to find an effect of probiotics on improving growth or reducing mortality. But I think the take home from that is probiotics have got a kind of dirty name, especially in the kind of medical world, but not all probiotics are the same. And there's some really exciting evidence for certain strains of probiotics which might potentially have some effect in these kids. And there was a big trial only a few years ago showing that a big probiotic strain was able to reduce mortality and sepsis in children in India, in a big trial of about 4,000 children. So there is potential for some of these species to work for growth and for infection in children from these areas. And there's also potential for other therapies such as nutritional ones which target the gut microbiota, such as those which are rich in fermentable carbohydrates to improve growth as well, but that's ongoing. So if we move to the next slide, the kind of final word on this is this potential of fecal microbiome transplants. Now, this is quite relevant in the kind of clinical gut world because these are the most effective therapies for treating C. diff infections in high income countries, whereby you take a stool sample from a healthy donor and transplant it into someone with recurrent C. diff. There's a 95% recovery rate compared to a 30-40% recovery rate from the previous standard treatment of antibiotics. So there's a couple of trials, pilot studies, looking at whether fecal transplants can work in severe cases of severe acute malnutrition in kids that aren't responding to normal treatments. And this is ongoing, but it is exciting to think that a microbiome targeted intervention like that could be a potential treatment for severe cases of severe acute malnutrition in low resource settings. you can go to the next slide. And that's everything. Yeah, thank you very much. And I'm happy to take any questions.